This video is for The Living Earth, Unit 4.2, DNA. Today's essential question is, what are the components of DNA and RNA? Our first main idea, DNA and RNA. Like other biological molecules, nucleic acids are large polymers made up of smaller repeating units called monomers. Nucleic acid monomers are called nucleotides. DNA contains 4 different nucleotides, each with a different chemical base: adenine, guanine, thymine, and cytosine. These bases can form hydrogen bonds with each other, but only complementary bases can bond. Guanine bonds only with cytosine, while adenine bonds only with thymine. RNA also contains 4 different nucleotides, 3 of which are identical to DNA bases. But in RNA, the base uracil replaces thymine. Uracil is therefore complementary to adenine. In addition to the bases, nucleotides also contain a 5-carbon sugar, which is called deoxyribose in DNA and ribose in RNA, as well as a phosphate group. The sugar and phosphate groups of adjacent nucleotides bond together to form a backbone. Scientists label the carbons in the sugar from 1' to 5' based on their position in the molecule. The phosphate group is attached to the sugar at the 5' carbon. The 5' end of one nucleotide bonds to the 3' carbon of the adjacent nucleotide to form the sugar-phosphate backbones. The 2 backbones of the DNA molecule run in opposite directions. One sugar-phosphate chain runs in the 5' to 3' direction, and the other runs in the 3' to 5' direction. DNA contains 2 separate complementary strands of deoxyribonucleotides held together by hydrogen bonds. The 2 strands wind together to form a spiral shape called a double helix. In contrast, RNA is made up of a single strand of ribonucleotides. Although RNA is single-stranded, however, some RNA molecules form folded structures, and, like proteins, these folded RNA molecules perform various functions, like catalyzing reactions and transporting molecules. DNA and RNA are very large molecules, but they follow the same chemical rules that apply to smaller molecules. For example, the charge on DNA's phosphate backbone is negative. This makes DNA a polar molecule, and it behaves like other smaller polar molecules, such as water. Because DNA is polar, it dissolves easily in water, and it does not dissolve in nonpolar solvents such as alcohol. DNA in eukaryotes and prokaryotes. In eukaryotic organisms, DNA is contained inside the cell nucleus. DNA is tightly coiled around proteins to form structures called chromosomes. This packaging enables the long, delicate strands of DNA to be compacted into a tiny space. It also provides a way for cells to control gene expression. The chromosomes of eukaryotes are typically rod-shaped. By contrast, the DNA of prokaryotes is usually arranged in a single circular chromosome that is located in an area called the nucleoid. Each eukaryotic species contains a certain number of chromosomes, each with a characteristic shape. Chromosome number does not directly relate to complexity of organisms, however. Chromosomes of sexually reproducing species come in pairs, one inherited from each parent. Humans have 23 pairs of chromosomes, or 46 total. A dog has a total of 78. And a particular fern, the adder's tongue fern, has a total of 1,260. At this point, you should be able to generate a full and appropriate response to today's essential question. What are the components of DNA and RNA?